Afternoon, everyone. My name is Wei Yuan Hsieh, and I am from the Division of Infection and Pathway Medicine. Before I start today, I would like to thank the Cardinal Memorial Committee for recognizing my work. And I will give you a very brief presentation on my talk, and the title is Metabolic Regulation of Viral Infection. So we've been interested in the interaction between host and pathogen, and particularly the macrophages in response to viral infection. And what I have done for the past 4 years is bring in another element, which is metabolism, in this case, cholesterol metabolism. And there are two key points I would like to share with you today. And the first one is I would like to show you that viral replication is dependent on the isoprenoid arm of this cholesterol pathway. But more importantly, cholesterol pathway is regulated by interferon through an oxysterol known as 25-hydroxycholesterol. So here's the model we've been using. We're using a virus known as cytomegalovirus, or CMV. It's a DNA virus that is part of the larger beta herpes family. And just like all the herpes, they're clinically relevant. So the transcription and replication of this virus is falling through, divided into different phases. So we have the immediate early, and late. During this process, the virus is required to interact a bunch of cellular factors and pathway for efficient replication. Although most of these viral transcripts are designated for the replication of the virus, more studies have shown that the CMV virus actively alters host metabolism pathway, including energy metabolism, or the lipid biosynthetic pathway for efficient replication. On the other hand, cellular factors or pathway can be internally or externally influenced or regulated by our immune system. And this inhibits the virus to progress and to complete its own replication cycle. And this model puts equal weight to both viral and host factor, such that viral replication cannot effectively progress without all the host changes having accordingly taken. So I'm going to focus on one subset of lipid pathway, the cholesterol pathway. I'll tell you how this pathway is required for viral replication and how this pathway is regulated by the immune system. So this is a cartoon version of the cholesterol synthesis pathway. It has 27 enzymatic steps. At the end, it produces cholesterol. And cholesterol is used to make cell membrane and hormones. Cholesterol pathway also contains side arms, which is known as the isoprenoid arm. And this is particularly important in vesicular transport, protein glycosylation, and to some extent, viral assembly. What's interesting about this pathway is regulated by a master transcription factor known as SRBP2 or SREP2. And this pathway has been described previously. It's been involved in replication HIV, HCV, and dengue virus. So given that this pathway seems to be important for the viral replication, when I first started, I asked the question, is this pathway regulated by interferon? So what we have taken is an unbiased approach of temporal exposure profiling of macrophages that are either being infected with CMV virus or stimulated with interferon. So just show you, over the course of 12 hours, we see a global downregulation of the entire pathway in the macrophages that are being infected with CMV. Interestingly, we also observe similar response of macrophages that have been treated with interferon over the course of 12 hours. So from this, we said that the primary response of infection is triggered the release of interferon. But to directly demonstrate that it is interferon that's regulating cholesterol pathway, we can use an interferon receptor knockout. In this case, in the wild type, we see a reduction in the pathway gene expressions. However, in the interferon receptor knockouts, this response has been completely abolished. And from this, we concluded that, yes, the cholesterol biosynthesis pathway is regulated by interferon. So knowing that this pathway is regulated by interferon, the next question I have to ask is, what's the dependency of viral replication on this pathway? So we can take an approach of using RNA silencing or RNAi to knock down enzymes that are on the pathways. So just quickly show you that if we knock down enzymes that are upstream of this isoprenoid arm, leading to the RAP protein pathways, leads to a significant inhibition of viral replication. Conversely, if we knock down genes that are downstream of this isoprenoid arm, it will lead to the metabolic shunting in the pathway, leading to a promotion in viral replication. So another approach what we can take in is using pharmaceutical agents. Statin is a class of cholesterol-lowering drugs that blocks the enzyme HMG reductase. As a result, this blocks cholesterol synthesis and therefore lowering cholesterol. What statin also can do is block the isoprenoid arm. And just to quickly show you it does that, we can use a marker known as unprenylate RAP1A, which is accumulated in response to statin treatment. And that shows that the isoprenoid arm is being blocked with the treatment statin. So, is statin antiviral? Here I will just quickly show you that when we treat a cell with statins, and in fact with CMV, we see a very significant inhibition in viral replication. What we can do next is to perform a metabolic rescue experiment by adding metabolites that feeds into different parts of the cholesterol pathway. So if we add things like vavelinate or gernyl, which feeds directly into this isoprenoid arm, we have a complete rescue of viral replication. Conversely, if we add squalene, which is turned into cholesterol, we see no rescue of the virus. And that shows that cholesterol itself is not what the virus is dependent on, but rather the pathway towards the isoprenoid arm is important. And this result is consistent with the previous RNA knockdown, showing the pathway leading to this generalization branch of the cholesterol pathway. So what does this arm do? So the isoprenoid arm of the cholesterol pathway generates lipid tail that can be attached to proteins. In this case, the RNA data shows these are the RAP proteins that are being attached. RAP proteins are responsible for transporting cargoes between specific cellular components inside the cell. And literature have described that pathogens such as hepatitis D actually uses this pathway to gain this tail for packaging. On the other hand, viruses like hepatitis C that actually requires a host protein to receive this lipid tail for efficient replication. So the question is, does CMV encode any proteins that require lipid tail modification? And we've done the bioinformatic analysis, and the answer is no. So it must be a host protein that requires this lipid tail modification for efficient viral trafficking. What we can do next is to perform a systematic unbiased screening of all 50 or so RAP proteins. And to cut the story short is we can identify a cluster of RAP proteins that are absolutely essential for efficient replication of the virus. So now that I show you that the virus is the pathway is under the regulation of interferon, and the virus depends on the pathway for replication. So how does interferon regulate the pathway? So let's go back to the transcription factor SRBP2. SRBP2 is known to be inhibited by oxygenated cholesterol, known as oxysterols. So I went back to our macrophage and asked the question, which oxysterols are produced by our immune-activated macrophage? So if we screen all the naturally occurring oxysterol in our immune-activated macrophages, only one 25-hydroxy cholesterol is being synthesized by this macrophage. And this catalysis from cholesterol into 25-hydroxycholesterol is catalyzed by the cholesterol hydroxylase CH25H. Here's just to quickly show you that this gene is rapidly induced when macrophages stimulate with the interferon or is in response to infection. So to cut the story short, is macrophage, once being activated, secretes 25-hydroxy. So is this 25-hydroxycholesterol antiviral? Here I give you two examples, one using a mouse system, so the mouse CMV virus, and the human CMV virus. Just quickly show you that 25-hydroxycholesterol can elicit strong antiviral effects against both viruses at a physiological relevant concentration. So to complete the story, what we can do is to take macrophage that are lacking this hydroxylase gene and infect them CMV. What we see is that macrophages that are lacking this hydroxylase gene are much more permissive to CMV infection. And since we published this work, other studies have shown that 25-hydroxycholesterol is effective against multiple pathogens, including flu, HIV, HCV, and dengue virus. So to go back to my first slide, I show you a three-way interactions between the host, the virus, and the cholesterol metabolism pathway. I've shown you that the regulation cholesterol biosynthesis is part of the interferon antiviral response. And from the RNAi knockdown, we show that there's a strong dependency of viral replication on the isoprenoid arm of cholesterol pathway. And that is one of the mechanisms is because this pathway directly regulates RAC proteins that are involved in trafficking of the virus. And lastly, one of the mechanisms in which interferon regulates cholesterol biosynthesis is through the synthesis and secretion of 25-hydroxy. And 25-hydroxy can elicit strong antiviral effect against multiple viruses. So here's a quick overview. What I have done in my PhD is connecting the circuitry between the workings of our immune system with the cholesterol synthesis pathway through the 25-hydroxycholesterol loop. 25-hydroxy leads to a suppression of the pathway, much similar to what statin does. This blocks the isoprenoid arm of the pathway, which rendering the RAP GTPases ineffective, and thus that protects the host from infection. So lastly, I would like to thank my supervisor, Peter Gazelle, who's in this audience. Also group members in the Gazelle group, Kevin Robertson, and Matthew Blond, and Kai. And I would also like to thank from Till Backman's group, Senthil Gandhi and Shadia Syed. And lastly, from Yaganha's group, Sam Griffiths, for all their help. And lastly, our international collaborators from Florida, California, Swansea, and Singapore. Thank you very much.